All right. Good afternoon, everyone. So today we have Dr. Stephen Bell from MIT. Dr. Bell is also a Harvard Hughes investigator. Dr. Bell is actually renowned for discovering the origin recognition complex, ORC in short, and his lab works a lot now on helicases involved in DNA replication. Today he's going to talk about molecular geneticism during eukaryotic DNA replication. So let's welcome Dr. Bell for today's biochemistry seminar.
Okay, so thank you, CJ, for that introduction. So my lab focuses on how eukaryotic cells duplicate their chromosomes. And we focus mostly on the earliest event in this process, replication initiation, because it's the most highly regulated part of the process. And what I want to tell you about today is a story concerning the very earliest event of replication called origin licensing, and how we've used single molecule approaches to gain new insights into the molecular dynamics of those events, including events that, as I'll preface my title, involve DNA bending, protein sliding on DNA, and protein leapfrogging other proteins, okay? So before I tell you about DNA replication, I want to make really sort of a pitch for why we decided to do single molecule studies to approach this series of events. And I think most of you, probably all of you, know that there are numerous events in cells where the cell needs to assemble complex molecular machines at specific sites in the cell. This includes events like transcription, replication, recombination, also something like vesicle transport or transmembrane signaling. All requires the assembly of these complex structures at specific places. And typically, I would say the primary approaches to understanding these assembly events are twofold. The first is to eliminate, either genetically or biochemically, a critical protein in that process and basically ask what doesn't happen, okay? And then the other approach has really become much more prominent due to the advent of cryo-EM, which is to determine the structures of key intermediates in these processes. Both of these are incredibly powerful. And we've learned lots and lots about these events through them. However, they both struggle to give an understanding of the kinetics and dynamics of these events, because in essence, for them to work, you have to stop the reaction, okay? Certainly true for structural biology, but also if this elimination is going to tell you something, it needs to make the reaction work differently, okay? So what we thought is that we could complement these approaches, and you'll see very much in particular, we complement the structural biology. Much of what I'll tell you today couldn't have been done without structural biology preceding it, is to use single molecule approaches so that we can look at the transitions between these various intermediates and the dynamics and unstable intermediates that are integral to these assembly processes. Okay, so if Aaron hasn't told you that before, I'm telling it to you again, okay? So that's my pitch for why you might be interested in this. Let me tell you a little bit about the events of replication initiation in eukaryotic cells. This starts during the G1 phase of the cell cycle, where two copies of the core replicative helicase enzyme, the MCM2 through 7 complex, are loaded onto origin DNAs. Now, while this marks every potential origin of replication, these helicases are completely inactive at this point, and this is a process called origin licensing. And they only become active when cells transition from the G1 phase of the cell cycle into the S phase of the cell cycle, where two kinases, DDK and SCDK, phosphorylate a number of proteins that then associate with the helicase and activate it. And the resulting unwound DNA plus the helicase can recruit the remainder of the DNA synthetic machinery to form a bidirectional pair of replisomes. Now, critically, it is the separation of origin licensing in G1 and helicase activation in the S phase that ensures that no chromosome can be replicated more than once, and indeed no origin can initiate more than once during the cell cycle, okay? And this is in the budding yeast Saccharomyces cerevisiae, where all the work I'm going to tell you about today was derived from, I should really say, it's not performed there, is mediated by the inhibition of origin licensing by the same enzyme that activates the helicase. So the transition from the state where you can't license your origin to where you can activate the helicases does two things. It turns on the helicase, but it also prevents any new events from happening. And this will become relevant at the last part of the talk that story I'll tell you there. Now, today I'm going to focus exclusively on origin licensing, as I mentioned before, so I want to tell you a little bit more about how this works. So this process involves opening the heterohexameric ring-shaped MCM2 through 7 complex at a specific interface between the MCM2 and MCM5 subunits, placing it around the origin DNA, closing it, and doing this twice such that the two helicases interact through their N-terminal domains, that is, they're in a head-to-head conformation. And this ensures that when the helicases are activated, you get them to pass one another. That makes sure every base pair is unwound at the origin, and it is also the first marker for the bidirectional nature of replication. So this juxtaposition of the helicase ensures that every origin initiates bidirectionally. And so understanding origin licensing is very important to understanding how replication is bidirectional. Now, these helicases don't load themselves. There's 3 other proteins that are required: ORC, CDC6, and CDT1. And these are the only 4 jumbles of letters and numbers you need to keep in your head for the talk today. I'm not going to make you remember all the later steps in the reaction, which involve probably a dozen other of those. Okay, so work from my lab and a number of other labs, including the Diffley, Stillman, Speck, Lee, and Costa labs, has generated a model for these events. And this model primarily comes from the 2 approaches I emphasized before, either eliminating proteins and seeing the consequences or determining structures of intermediates. Okay, and the first step in this process is ORC binds a conserved sequence found at every yeast origin called the Ars consensus sequence, and then it recruits a copy of the CDC6 protein, which completes an ORC CDC6 protein ring around the DNA. This in turn recruits an open MCM2 through 7 ring bound to CDT1, and in fact CDT1 is important to hold it open, and it places that ring around the adjacent DNA to the ORC binding site to form an early intermediate that has all 4 proteins that's referred to as the OCCM because it has ORC, CDC6, CDT1, and MCM in it. Okay, then in a dramatic transition, instead of ORC binding to the C terminus of the MCM, there is a subsequent complex in which it binds to the N terminus of the MCMs. And I'm going to tell you a lot more about this transition in a little while. But this complex can then recruit a second CDC6, a second MCM CDT1, and then loads the second MCM on the DNA, again, adjacent to where ORC is, and this results in the head-to-head assembly of the 2 hexamers, okay? Now, as I said, this comes from the 2 traditional approaches, and indeed, there are structures of all of these intermediates that have been determined by cryo-EM. But what I really want to focus on today is what goes on in between these events, okay? Particularly between this OCCM to the MO complex. You can see that it would not be easy to simply morph between these 2 structures and understand the events that happen, okay? So how do we do these single molecule experiments that I'm going to be telling you? We use a process called co-localization single molecule spectroscopy, or COSMOS, that was originally developed in our collaborator Jeff Gellis's lab. And put simply, what we do is to tether origin-containing DNA on the surface of the slide, at the opposite end of which is a blue fluorophore. We then perfuse into that chamber the 4 purified proteins required for helicase loading, at least one of which, and typically 2 of which, are labeled with different colored fluorophores. We then use turf microscopy to illuminate the volume immediately above the surface of the slide and use the co-localization of protein and DNA fluorophores as a proxy for DNA binding. Okay, many of you may have heard Aaron describe similar types of experiments. Now, one thing that's not illustrated here, but which I will use extensively in this talk, is that very frequently these red and green dyes are positioned such that they can perform fret in different circumstances so that we can monitor specific protein-protein or protein-DNA interactions in these real-time observations. Okay, so how do we analyze this data? We simply identify all the regions that DNA is attached in the field of view, and then after we collect the images of fluorescent proteins associating with this in a post-hoc state, we can basically ask when do proteins associate with a specific DNA molecule, and we use this to generate traces that look like this, where we can see both very short-lived associations as well as much longer-lived associations. In this case, this is showing the association of a first and a second MCM molecule with the DNA. And you'll see a lot of things like that in the talk. Okay, I want to start by telling you a little bit of historical work done by the first student, and actually everything I'm going to tell you about today is done by graduate students. I've actually never had a postdoc work on this project in my lab. Okay, and it was started by a very brave student, Samina Takao. We had no experience doing single molecule work. She said, I'll do this. And all of this is done in collaboration with Jeff Gellison, a senior research scientist in his lab, Larry Friedman. They not only build the microscopes that we do all the experiments on, but they are integrally involved in the subsequent analysis of the data. Best collaborators ever. Okay, so the first thing Samina wanted to do was to just pairwise put the MCM proteins with each one of the helper proteins, ORC, CDC6, and CDT1. And on the left, you'll see an experiment with CDT1 and MCM, on the right, CDC6 and MCM, and they probably look a lot the same to you. In each case, we see a short-lived association of the CDT1 or the CDC6 when the MCM complex is recruited, okay? The only difference, which is very hard to see here, is that CDC6 always anticipates the MCM arrival, whereas CDT1 arrives with the MCMs. But the thing we were sort of interested in is when they leave, okay? Was there an order to these events? This is something that's very easy to do in these sorts of experiments. And so Samina plotted on this so-called survival plot. She synchronized all the events relative to the arrival of the MCM. At that time, 100% of the molecules have CDC6 and CDT1 because they are forming this OCCM complex. And then she asked, at various times afterwards, what fraction of the molecules still had CDC6 and what still had CDT1. And I hope it's clear to you that on average, CDC6 leaves before CDT1, and I will tell you, but won't go through the data, that this is true on a single molecule basis. CDC6 always leaves before CDT1. When she found this, we thought it was really interesting. We had no idea what the importance of it was, okay? But I'm gonna explain to you what that is as the talk proceeds. Now, she also did the same experiment with MCM and ORC and got a very different observation. And that was she saw a single ORC bind, recruit the first and the second MCM, and then rapidly come off the DNA again. And this is just two examples that are very similar to one another, okay? And that was surprising because how does ORC load two MCMs in opposite directions, okay? You've seen a little bit of the answer to that, but when we originally got this observation, it was quite baffling to us. And in fact, Samina went on to work on many other things in the lab, but she left this problem for a later graduate student, okay? And that student is Shalini Gupta, and she wanted to try and understand how could one ORC load two MCMs, okay? And basically, when Samina left, we had this very interesting model. We suggested that the way one ORC could recruit two MCMs is it would form this initial interaction. And at this time, the structure of the OCCM was known, but that MO complex structure was not, okay? But that the second MCM would be recruited by N-terminal interactions that are seen as very tight associations in the final double hexamer. We thought this was very clever. It turns out it's definitely wrong. Okay. At the same time, the Diffley lab, based on studies showing that each origin had to have both a high affinity ORC binding site in one orientation and a second ORC binding site in the opposite orientation in order to get helicase loading, they came up with a 2-ORC model where one ORC binds at one site, one binds at the other, and they sequentially recruit the 2 MCMs, which somehow slide together to form this structure. Okay. So Shalini thought, okay, I should be able to distinguish between these 2 models, and a good way to do that would be to design a FRET probe that would detect the interaction observed in the OCCM, okay, which is this C-terminal interaction between the C-terminus of ORC and the C-terminus of the MCMs. In the 1-ORC model, we'd expect to see first 1 ORC present, and second, only a single round of this interaction, okay, based on this top model. In the 2-ORC model, we do not only expect to see 2 ORC binding events, but we'd also expect to see 2 of these interactions. Okay. As is often the case in these huge field controversies, everybody's wrong, okay? So what we observed was, let me just take you through the data. So the first thing is, here's the first MCM, here's the second MCM. That's really easy to see here. The second thing is, this is 1 ORC getting on the DNA, and all the variability here is due to the decrease in the green signal when you see FRET. Okay, and I'll tell you a little bit more why we know this is a single ORC in a moment, okay? But what was surprising is that clearly, even though there's 1 ORC, you're seeing 2 rounds of this interaction between ORC and MCM, okay? So 1 ORC, 2 interactions, not 1 ORC, 1 interaction, or 2 ORCs, 2 interactions, okay? So what we can conclude from this is that ORC uses the same interface to recruit the first and the second MCM. And what I'm not going to take you through, because it's very complicated but immensely clever set of experiments that Shalini did, where she labeled ORCs with different fluorophores and was able to demonstrate that the same labeled ORC always loaded the first and the second MCM. And that's really the smoking gun evidence that it's the same ORC. If it was going out into solution, it could exchange the fluorophore association, and you'd expect to see different ORCs doing the first and the second, okay? So 1 ORC does both these things. We know that both binding to this site and binding to the second inverted ORC binding site is essential for the reaction. And what that led us to is a model where ORC must somehow release from its initial binding site and rebind in its second binding site. Okay, and when this happened, when we had this data, we basically didn't know about this second complex, this inverted complex, okay? So these are the 2 structures. This is the OCCM. This is done by the Speck, Stillman, and Lee labs. This is the MO complex that's done by the Costa and Diffley labs. And I just want to take you a little bit through this. So this is this C-terminal interaction. You can see the extensive interface between ORC and MCM here. And I'm going to call these OM interactions to distinguish them from the opposite sort of orientation of the 2, which I'll call MO interactions. And here, ORC is bound to the N-terminal face of the MCMs. And interestingly, it does not use this large C-terminal interface, and instead uses a single protein called ORC6 to make these interactions. And interestingly, this is the only protein that's not involved in DNA binding and does not encircle the DNA. Okay, so Shalini thought this is really cool because maybe this is somewhere along this pathway to switching binding sites. And so she wanted to monitor this in the same way that she did the OM interactions. So she created a new FRET pair that would have FRET when you form the MO complex, but would not show FRET in the OM interactions. Okay, so she did that experiment and got very different results in terms of the FRET. So in contrast to the OM interactions where we saw it started right as soon as the MCM arrived, here there's a significant delay, and then you see the MO interaction. And interestingly, while it never coincides with the arrival of the first MCM, it is always present when the second MCM arrives, okay? So we never see a second MCM recruited when an MO complex isn't there. And this is just the same thing with a different molecule. Okay, so what we see is there's only one MO event. The MO complex forms after the arrival of the first. It's always present for the second, and it dissolves well before ORC comes off the DNA. So that's why you see this nice dip and coming back up in the green signal here. Now, one thing we were interested in was the transition from the formation of the first MO complex, which of course happens immediately on the MCM arrival. Okay, that's the formation of the OCCM, and then the formation of the MO complex, because these two things are not compatible with one another. Okay, you can't be in the MO and the OM at the same time because ORC is on opposite sides of the MCM molecule. So one of the great things about single molecule studies is to be able to determine the kinetics of various events. And what Shalini did was to measure the time of the end of the OM interaction here versus the time of the formation of the MO interactions. She did this relative to several different events, but I'm just going to show you one, which is CDT1 release. Okay, so I showed you earlier there's an ordered release of CDC6 and then CDT1, and it turns out this is the one that coincides most temporally with the end of the OM and the beginning of the MO. So if you look first at the data where we measure the time of the breaking of the OM interaction and the time of CDT1 release, we basically are subtracting these times from one another. And what we see is that they center around 0, suggesting that they are either simultaneous or tightly linked with one another temporally. Okay? But if you look at the formation of the MO complex, all the numbers are positive, and what that means is that the MO complex is forming after CDT1 is releasing, but very quickly, within about 4 seconds, the majority of these complexes have changed from one orientation to the other. Okay, so what have I told you so far? So I've told you there's ordered release of CDC6 and CDT1, that the release of CDT1 breaks this interaction between ORC and MCM, and then very quickly afterwards, you get into this new conformation, presumably by ORC flipping over the first MCM. Okay, so we have questions, right? 2 important questions. The first is, why does ORC ever release from this high affinity binding site that it initially binds to, right? For it to do this flip, it has to let go of its original binding site, flip over the first MCM, and actually bind to a lower affinity binding site. So why does it do that? And then the second one is, how does it do that without being released into solution? Okay, there's got to be something tethering it to some part of either the DNA or the proteins bound to it to keep that from happening. So I'm going to start with the first question. Why does ORC release from its initial strong binding site? And this actually, we answered this question a little bit inadvertently. This is the work of Annie Zhang in the lab, and she was primarily looking at the bending that's induced by ORC DNA binding. Okay, and this stemmed from 3 structures, all of which were determined by the Speck, Stillman, and Lee labs in different papers. The first is showing the bent DNA when ORC binds it. This is showing it with CDC6 here in the purple, but you see the exact same thing without CDC6. And you see this very robust 80-degree bend in the DNA that's consistently observed when ORC is binding to it. The second one, they captured what they believed to be an intermediate in the MCM recruitment by mutating part of the MCMs that had a partial interaction with ORC, but which the DNA is still bent. And interestingly, that DNA sits right over the opening in the MCM 2 through 7 ring through which the double-stranded DNA needs to go to enter into the central channel of the MCMs. And then this is just another version of the OCCM complex. And as you can see here, the DNA is straightened. It's going straight through the middle. It's basically a linear piece of, a straight piece of DNA that goes through the center of both of these ring-shaped complexes. So what Annie wanted to know is, first of all, is this bending really as static as it appears to be in the structure? Okay, is it actually bending, unbending, bending, unbending, or is it just always this way? And does it impact ORC's affinity for DNA? There's actually not a lot of interactions out there, obviously enough to induce the bend. And then finally, what regulates the straightening process? Why does it actually go from this bent state where it's obviously interacting to this straightened state where it's been deposited? And to begin to answer these questions, she started with a simple protein DNA fret probe. She labeled the DNA such that when it was bent, you would get a high fret interaction with a donor attached, in this case, to the ORC6 protein, but when it was straight, you'd see low fret. And the first experiments she did were really simple, just ORC and the DNA on the slide. And what she saw in the vast majority of cases, every place where that's outlined in blue here, is that as soon as the ORC got on the DNA, you saw high fret, and that stayed until it fell back off, okay? But occasionally she saw this, where it got on, you saw high fret right away, but just before it fell off, you saw a low fret signal. The fret signal dropped back down. ORC is still on the DNA because we still see a green signal. Now, if she takes all this data and plots it on a single plot, she gets a very big peak at the high E fret interaction state. That's consistent with it mostly being in that case. Those of you in the front row might see that there's a little bit of data down here, but if I zoom in, you can see there's actually a reasonable amount there. And so what Annie, being a good single molecule scientist, did was to ask, was there different properties of these two different populations? So she asked this by asking, was the off rate different when you were in the low fret state versus the high fret state? Which is more or less asking, are you in a higher affinity state when the DNA is bent or when it's straight? And the answer was really clear. It's about a 10-fold higher off rate if you're in the low fret state than if you're in the high fret state, okay? Now, she did another experiment similar to this, but instead of changing whether the DNA was bent or not in her observations, she actually did these same observations with and without CDC6. And you can see this does very little to the E fret values that we observe. That's consistent with the structure, which sees the exact same bend in each case. But if we look at the off rate of ORC without in black or with in purple, you can see that release of that CDC6 enhances its binding for DNA, okay? Now I'm gonna take you back to the order of events. You'll remember that what we observe is that the DNA has to straighten in order to enter the MCM channel, and CDC6 is the first protein that comes off. So she knows two events already that are reducing ORC's affinity for its original binding site relative to when the reaction starts, okay? Okay, she also wanted to know, when does the straightening occur? I'm not going to take you through how she changed this out for a quencher, but suffice it to say, after the MCM arrives, basically 10 seconds later, almost all the molecules have straightened their DNA. We wanted to know, is this associated with deposition? That is, it looks like if the DNA straightens, it will drop down into the MCM central channel, but do we actually see that? And to look at that, she just adapted this DNA the DNA probe to donor that was at the end terminus of the MCM. So in this case, if the DNA is bent, we'll see low fret. If it's been deposited into the central channel, we'll see high fret, okay? When you look at these traces, this is just showing MCM in green, and here's the fret value. You see very quickly after the MCMs get on the DNA, you get a high E fret value, okay? And this is just showing one molecule, but I can show you that if we look at the timing of that with many molecules and we compare it to the time of DNA unbending here, you see that the time of DNA unbending and the time of DNA deposition, the distribution sits right on top of each other, suggesting that these events are either simultaneously or occurring or tightly coupled to one another. Okay, now I snuck something by you in the last slide. I'm pretty sure no one will have noticed this. But if I take a bunch of the experiments and put them together and show you what's called a heat map, you'll see it, okay? So let me just explain what this is first. This is E fret values on the y-axis, time after MCM arrival on the x-axis. And this blue to yellow continuum represents the probability that a specific value is present at any given time point, with yellow being higher probability and blue being lower, okay? This is the same deposition probe, and consistent with what I told you before, all of the molecules are at this low fret value when they arrive. That's consistent with the bent DNA. And very rapidly, they transition to the high fret state. But the thing that you're not missing now is that we always see that fret value decay over time afterwards, okay? And there are many possibilities as to what this could be, but the one that Annie was most excited with is that maybe the MCMs were sliding away from their original site of DNA deposition over time. Okay, so she wanted to test this, and to do that, she made 2 additional DNAs with donors at different positions on the DNA. So this is the +51 is what I've been showing you, but because of where ORC binds in the DNA and the size of the MCM complex, this should essentially always show high fret upon initial deposition of the DNA, okay? But if you go to the right or to the left and slide, we should see fret with the +82 or the -4. And that's just illustrated here. So here's what you would see with the +51 when you initially deposit. If you move to the right, you're going to see the +82. If you move to the left, you'll see the -4. And I just want to emphasize what I'm going to show you the results of. It's just a bar graph, but it's 3 different experiments. We can't put these on the same DNA because the fret looks the same no matter which molecule we're interacting with, okay? But what we see is this. So for the +51, consistent with almost every molecule having the DNA deposited, if it arrives on the DNA, that is a very high percentage. But if we look at -4 and +82, we see that basically half the molecules go that way, okay? And this is much more consistent with sliding than, for example, translocation. MCM is a helicase. It, in fact, can translocate on double-stranded DNA, but it would go only towards the +82 if it were translocating the way it does during helicase activity. Okay, so we think this is very strong evidence that there is sliding. Interestingly, while we were trying to get this experiment to work, Annie managed to make 6 different MCM ATPase mutants. And when she tested them, she actually got a very interesting observation, which is that no matter which one of the ATPase motifs, there's 6 of them in the MCM complex, she mutated, what you saw is that they were more or less stuck at the +51 position. We didn't see the decay anymore, okay? So if it's not translocation, what is this? There's actually pretty good evidence that there's 1 round of ATP hydrolysis that is observed during helicase loading. And what we think this does is it goes from the ATP to the ADP state, and that disengages a series of hairpins that are part of the helicase mechanism such that it is no longer engaged with the DNA. And we think that allows MCMs to enter a sliding state at that point. And if you don't hydrolyze all 6 of them, apparently you still have enough engagement that you delay or eliminate. This one's, I think, a little bit delayed. The rest are sort of not sliding at all. Okay, MCM sliding on DNA, not new. At least 2 other labs had shown that they could observe MCM sliding on DNA. What they had never observed is when this happens during any event in DNA replication. And so we wanted to find out, when is this happening? Is it happening randomly? Is it happening at specific times? Okay, so Annie measured the time of the initiation of sliding versus the release of the CDC6 and the release of CDT1, okay? So I told you I was going to tell you that there's a reason for the order of these events. And what I'm going to show you here is that this, the plus values here indicate that it's happening, sliding is happening after CDC6 release, and in the majority of cases before CDT1 release. And we were particularly interested in this result. And that's due to the fact that I've told you that CDT1 release coincides with breaking the ORC-MCM interaction, okay? So if sliding is happening before CDT1 release, that means not only are the MCM sliding, but ORC has to be sliding along with it, okay? So Annie wanted to test this idea. She made an additional fret pair. In this case, the donor's on ORC and the acceptor's right next to the ACS. So if ORC is bound at the high affinity binding site, we'll see fret, okay? You look at the data, consistent with the ORC initially binding to the ACS, we see very high fret. As soon as ORC gets on the DNA, we actually see a very low green signal because most of it's going to the red fluorophore. Then you can see, here's the recruitment of the first MCM, and the fret stays low. But then at some point after that, ORC is still on the DNA because we still see a green signal, but the fret goes away. It's actually moving away from the ACS site, okay? And importantly, this always happens after CDC6 releases, and it never happens before CDT1 releases. Okay, so what does this mean? Let me just take you through how we think about this. So we know ORC binds and bends the DNA. We know the MCMs arrive. We actually know why, or at least the parts of the MCMs that drive this release from the bent state to the straightened state. There's 2 winged helix domains on MCM4 and MCM6. You eliminate those, you never see unbending of the DNA, you never see deposition. So we believe there's new interactions that are formed in this top part that drive the release of the bent DNA and deposition into the ring, okay? Now, at this point, there's sort of a gradual reduction in affinity for the ACS. The straightening of the DNA reduces the affinity. The release of CDC6 further reduces the affinity. And we think the last step in this process is the ATP hydrolysis by the MCMs, which disengages the MCMs from the DNA, allowing the whole complex to slide, okay? And what we find most satisfying about this means that at the time that CDT1 is released, remember, it's sliding before CDT1 is released, and you break this interaction, that means ORC isn't bound to its ACS anymore. It is on non-specific DNA, which will prime it to be happy to release from its initial site and initiate the flipping process, okay? So we think that order of events of CDC6 and CDT1 is critical to order the release of ORC from its binding site with the breaking of this initial ORC-MCM interaction. Okay, so that's the first question. What about how does ORC switch binding sites without releasing into solution? So that's the work of David Driscoll, another graduate student in the lab. And David really started his work by looking at the structure of the MO complex. And he was focused on what ORC6 was doing in that complex. In that complex, the N terminus of ORC6 is interacting with MCM2, the C terminus is interacting with MCM5, and it'll be important later for you to keep in mind that it holds the MCM25 gate closed, okay? And in the case of the C terminus of ORC6, it is bound very tightly to the body of ORC. There is no chance that that part of the interaction could occur before ORC releases from the DNA and flips entirely over, okay? But the N terminus, which is out here at the very end of the molecule, is at the end of a 165 amino acid linker, more than enough distance to reach over and bind to the other side of the MCM complex, okay? So we were interested in the possibility that this formed a molecular tether between ORC6 and the N terminus, and that was holding ORC as it did its flip, okay? So David made a fret probe where he could monitor the interaction of ORC6 with the N terminus of the MCMs. We knew that would be present when you form the MO complex, but what we wanted to know is, did that interaction occur before the MO complex? Was it anticipating that more robust change in ORC location? So David took that probe and put it into the reaction. Here's ORC binding to the DNA. Here's the first MCM getting on, same thing over here. And you can see very rapidly after the MCM is recruited, we already see this interaction, okay? Well before even the dissolution of the OM complex, and certainly earlier than the MO complex is forming, okay? If I show you many examples of this, this is the same kind of heat map, where we're measuring from the time of MCM arrival, you can see within about 10 seconds, you see a transition from the low fret state to the high fret state, and it's more or less permanent. So there's a strong interaction formed and it's maintained. Okay, now some of you may be saying, well, you have a donor fluorophore on the end of a 165 amino acid thing. Maybe it's just floating in the right vicinity, so this isn't a specific interaction. And although the reviewers didn't ask us this, we anticipated it anyway. And what we did was to do a different MCM complex where we had moved the acceptor fluorophore from the N terminus to the C terminus. And you can see when we do that, not only do we not see the transition to the high fret state, we actually see a reduction in fret in the same time frame, presumably recognizing that instead of floating around, we've now pinned that domain far away from where the fret acceptor is. Okay, so we really think this is a specific interaction. The real clincher in this is if we make mutants in the MCM2 region that binds to the ORC6 N terminal, we completely eliminate the interaction. If we mutate either by deleting the N terminus or by mutating the linker region adjacent to it, both of them have effects on this. Interestingly, deleting the N terminus has the weakest effect, and that we believe is because this linker region also interacts, and in fact, in the absence of the N terminus, that can occur. But if we delete both, we completely eliminate that. And if anybody wants to know more about the complexities of the ORC6 mutants, I'm happy to tell you later. Okay, so we think we know the tether, but does it actually happen at the right time, and does it have the right effect? Okay, so what we did is to first ask, is the tether present before CDT1 releases, okay? Before CDT1 releases, we aren't really worried about ORC wandering off because it's bound to the MCM complex, but it should be tethered when it releases so that if it does come off, it's retained at the site of origin licensing, okay? So David did a simple experiment and asked, relative to CDT1 release, what percentage of the molecules have already formed the tether interaction? And the answer is, I'm not going to take you through the math here, but 151 out of 152 molecules we looked at had formed this well before the tether, before the CDT1 is released. So it's happening at a time that's relevant. Does it actually affect ORC retention? Okay, so there's really two fates of ORC after CDT1 is released. One is it can come off the DNA entirely, float away. If this happens, we actually know that the MCM rapidly is released, presumably because it has an open gate and it simply falls off the DNA. And the other is what I've really shown you throughout the talk, which is that it actually goes on to form, it's retained, it goes on to form the MO complex in a double hexamer. So David asked whether the tether affected these two fates in two different ways. The first way is he looked at the time of CDT1 release, whether a particular ORC molecule had or had not formed the tether by it being in either the high fret or the low fret state. And then he asked, how long does that ORC stay around after that point in each case? Okay, so this is again a survival plot. All of the molecules have ORC and CDT1 at the time of its release. If you have low fret at the time, you have much shorter dwell times than if you have a high fret at the time, indicating that the tether is improving the retention of ORC on the DNA. We can do the same thing with the ORC6 mutants that I described earlier. And despite the fact that some of the linker ones have, the N-terminal mutant having a relatively small effect on the fret, what I'll tell you is that if we delete the N-terminus and measure the ORC dwell times after CDT1 release, you see very, very short dwell times of ORC on the DNA, okay? And if you delete the N-terminus and the linker, you see very similar things. Okay, so we think that we understand both of these questions. We think this ordered series of events allows ORC to come off the DNA, and then the ORC6 tether is holding it there. So I want to take the last few minutes and take the opportunity to tell you one last story, which I think points out that some of the things we're talking about are really biologically relevant because they are regulated, okay? And this is actually the work of Audra Amasino, graduate student in the lab who's now still working in the lab as a postdoc. Some of you may recognize the name, some of you may recognize the person, some of you may not recognize her, in which case maybe this is more helpful. So this is not when she was working in my lab, maybe working in some lab around here. And what Audra wanted to do was to understand how ORC phosphorylation inhibits origin licensing. So I alluded to this earlier that the CDK modification of proteins involved in origin licensing inhibits that reaction outside of S phase. And we've known for a very long time that CDK has 3 targets among the licensing proteins. It modifies MCM2 through 7, causing its nuclear export. It modifies CDC6, causing its ubiquitin-dependent proteolysis, and it modifies ORC. Interestingly, that's on ORC2 and ORC6. This is the only modification that intrinsically inhibits the reaction. The modification of CDC6 and MCM, if you're just doing it in a 4-protein reaction, no effect at all. You need either a nucleus or a ubiquitin proteolysis system for those phosphorylation events. Although that's true that we knew it was involved in the, it could be done in the intrinsic reaction, we actually didn't understand how it worked. Very early studies showed that you could form the OCCM with phosphorylated ORC. That's measurable in ensemble reactions. And you couldn't form the double hexamer, but those are the only 2 things you can measure in ensemble reactions. So there was a large variety of candidates for things that could be inhibited by phosphorylation. Okay, so Audra started these experiments by doing a very simple experiment. She just took labeled MCMs and did a loading reaction with either phosphorylated or unphosphorylated ORC. Okay, that was already very informative. If you have unphosphorylated ORC, you see a first and the second MCM loading, as I've shown you many times today. But if you have phosphorylated ORC, you only ever see first MCMs. They never successfully recruit a second MCM. Okay, so, and this is just showing 2 traces, but if we quantify this, we can show that about 50% of the molecules, first MCMs, when ORC is unphosphorylated, go on to form a recruit a second, whereas when ORC is phosphorylated, 0% do that. Okay, so that allowed us to eliminate these 2 potential sites of inhibition because we knew that you weren't even getting to this point. What I'll tell you but won't show you is Audra went on to show that it didn't affect CDC6 release, it didn't affect CDT1 release substantially. But there was one thing that she saw, and if you look carefully at the traces, you would have seen this, which is that whether ORC was phosphorylated or not affected the time MCMs were retained on the DNA. So this is in blue, phosphorylated ORC, in red, unphosphorylated ORC, and this is how long after the first MCM arrives it stays on the DNA. And you can see when ORC is unphosphorylated, it's much longer than if it's phosphorylated. And when Audra saw this data, she immediately thought of whether the ring of the first MCM was closing or not. And in fact, one of the things Samina did after she did the original experiments I showed you was develop a probe that could monitor whether the MCM ring was open or closed in real time. Okay, and so this is just a fret pair on the MCM2 and MCM5 subunits. When the ring is open, we see low fret. When it's closed, we see high fret. If you look at this data with unphosphorylated ORC, you see that most of the molecules, actually almost all the molecules start out in this open ring state, and then around 50 seconds, they transition and are retained in the high fret state. That is, they go from an open state to a closed state, and they stay in the closed state after that. Okay, there's not a lot of opening and closing at that point. When Audra did the same experiment with the phosphorylated ORC, she saw a very different result. The molecules mostly stayed in the low fret state, that isn't with the open ring, and only occasionally had brief forays up into the closed state. And what we think this means is they are capable of closing, but they are not being held stably in a closed state. And one of the reasons we think that is that we knew that the OCCM complex has an open gate, but once you get to the MO complex, ORC6 binds across the MCM25 gate and holds it closed. Okay, so what we hypothesized is that phosphorylation would prevent that interaction such that the MCMs could close, but they wouldn't stay closed because they didn't have the formation of the MO complex to do that. Okay, and having come to this hypothesis, she went over, Audra went and talked to Shalini and said, if I give you phosphorylated ORC, will you check whether the MO complex forms? And Shalini did that experiment, and it was not at all ambiguous. If you phosphorylate ORC, you see absolutely no MO complex formation. And we think this is what holds the ring closed and stabilizes the MCMs on the DNA. Okay, and the last sort of coup de grace of this is that David, when he developed his tethering interaction, said, hey, ORC6 is one of the proteins that's phosphorylated by CDK. Maybe it affects the tether. Okay, so he used his FRET probe and asked whether phosphorylation of ORC affected this. And you can see here's formation of the tether with unphosphorylated ORC. Here it is with phosphorylated ORC. Again, this is very similar to what I was describing with the ring opening and closing. And what he sees is very short periods on individual molecules of association, but nothing stays tethered. Okay, so we think what this tells us is that ORC phosphorylation is actually acting to inhibit the tether formation, and that has the downstream consequences of preventing this flipping reaction and therefore preventing MO complex formation. Okay, so that's that. I'm just going to end by taking you through the model and putting everything together. So I've shown you that we know that when ORC binds DNA, it bends it, and that's true when CDC6 joins it. When the MCM-CD21 complex joins, we think that not only does its interactions cause the loss of the bending, okay, and the deposition of the DNA, but right away, you have this secondary interaction of ORC with the opposite side of the MCM complex. And I sort of think of this as ORC putting its arm around its friend, right? So there's a major interaction here, but there's another interaction over there. Might even be ORC pulling a prank on its MCM friend. I'm tapping your shoulder on the other side. Okay, and at this point, we can then release CDC6, which further reduces ORC's affinity for the ACS. That then follows with MCM's hydrolyzing ATP, allowing this entire complex to slide around, pulling ORC off of, or at least allowing ORC to leave this original ACS binding site. When CDT1 releases, we break this interface, and because ORC is bound to nonspecific DNA, it comes off the DNA. Okay, now in work I haven't had time to tell you today, we actually know the next step is the formation of the protein-protein interactions associated with the MO complex, not the DNA interactions. So we can show that binding to the second site comes well after the formation of these initial MO interactions. And we think this represents something we're calling the pre-MO complex that can slide along the DNA and search for these B2 sequences, because they are not found at exactly the same position at every origin. In fact, there's up to, they can be as much as 400 base pairs away from the ACS. So this has to be found by ORC, and we think this complex allows that search to occur. And then the DNA can bend. You can recruit the second MCM-CDT1, CDC6 and MCM-CDT1 complexes, and then the sequential release of CDC6, ORC, and CDT1 allows you to form this final complex. Okay. I realize that this is my little fever dream, okay? And I don't expect you to remember all this. But what I hope you can understand is there are many, many aspects of this model that would have never come out of a cryo-EM structure, okay? The order of CDC6 and CDT1 release, nobody's ever seen a complex that doesn't have CDC6 and CDT1. They're either both there or neither there. Okay, this regulation of the tether, that tether interaction has yet to be described in any structure, okay? I think that's because there's an ATP hydrolysis dependent step and nobody's ever caught the right period where the right number of ATP hydrolysis have occurred, okay? The whole regulation by phosphorylation, if we had just done that, in fact, that's how we started, and only knew about the structures, we'd just say, oh, you can't form the MO structure, okay? But we see these new things. So what I really want to say is, while structural biology is awesome, none of what I showed you today would have been possible without the structures. All those FRET probes relied on the structures. It's a beginning, not the end of the understanding of these things, okay? So if you take one thing away from my talk, take that. I'll just end by thanking Audra, David, Shalini, Samina, and Annie. It's been one of the most fun periods in my lab, and particularly because it's all been graduate student. I'll thank Jeff and Larry, without whom none of this could have been done. They're the most amazing collaborators, and they look evil and mean here. They are the nicest guys you've ever met. And then the lab pups who keep us all sane, and our funding sources. And I'll take any questions.
So wonderful talk. It's amazing to see all that put together after so many years.
Yeah, I should point out this started in like 2013.
So the sliding, you have it going in one direction where, like on the way on the slides, the weak affinity site is to my right.
Yep, and that's typically true at origins. It's typically mapped on that side. But I can tell you it can work if you put one on the other side.
That's my question.
Yeah.
Okay.
So if you put one on the other side, it tends to not work as well.
Mhm.
And that may mean there is some predisposition to go one direction versus the other, but it will function. And if you think about the model, it should function.
And I don't know about like nucleosomes at origins. Like, do those form barriers?
So this is a fundamental flaw with everything I've shown you. There are no nucleosomes on our template, but they would restrict the sort of search space, right, for these things. And they probably do restrict which B2 elements can be used. One of the things about origins, they're very AT rich. One of the things about the things that ORC binds, they're very AT rich. And in fact, these weak binding sites just need basically 9 out of 11 base pairs to be A or T. So it's a very low bar to be a B2 element, but we can show your prediction is correct, right? So it's actually tricky to get rid of B2 elements at origin. So we put the ACS in a GC-rich template out of Myxococcus, and then we can put AT-rich places all over, and it will work in either orientation. Or sorry, either side, not either. It has to be anti-parallel. It will not work at all this way, but it will work here or here.
Hey, that was a great talk. So I've always been interested in the idea that origin licensing was considered so rigid, and the sliding idea is, I think, really interesting. Have you ever, I know Aaron just asked about nucleosomes, but have you ever also thought that maybe this lends possibility to sort of a DNA damage tolerance ability where, you know, it's looking to start an origin or origin fire. It can't line up exactly, and gives it some sort of ability to be a little bit more flexible?
If you're talking about where the MCMs end up, that's clearly true. So for example, you can set things up where you put an origin in the middle of a transcript, and you can show that transcription will push the MCMs around, but they'll still be used to initiate. So one of the things that's interesting and very different from, for example, bacterial replication, where there are sequences that are important not only for putting the helicase on the DNA, but actually initiating or unwinding the DNA. That's not true in the eukaryotic situation. It appears that if you get the MCMs on, they can initiate at any DNA sequence they're surrounding. So I think there may be some element. I'm not sure how that might be connected to repair.
Not necessarily repair, but just like tolerance of surrounding lesions or things like that.
Yeah, no, I think it would be very tolerant of that. And in fact, I even think the initial unwinding of the DNA would as well, unless it was extensively modified where the MCMs couldn't get any grip. The idea for initiation right now is that the MCMs interact with opposite strands of the DNA, so they will engage. We know that they can translocate on double-stranded DNA, but when they do so, they're only engaging with one of the strands, not the other. And when they do that and they are activated, they actually pull the two strands apart from one another. So they shear the DNA to do the initial unwinding. And then there's a magic trick where they send one strand out, the one they're not engaged with, and then they can bypass each other. And that all has to do with a protein called the MCM10, which is still a little bit mysterious what it does. So happy to talk more about that, but didn't talk about it today.
Any more questions? You have one. So for the OX6 linker, right? So you're saying like the flexible linker also is attaching to these.
We think it has an interaction. Interesting, there is data from a previous paper in my lab that it actually binds CDT1, which would be very cool because when CDT1 is released, you might change the nature of ORC's interaction. One of the things we see is you can imagine when we're doing that ACS interaction assay, that ORC could slide off of it and then come back and rebind the ACS. And we see that, but we never see that after CDT1 releases. There is something that is discouraging ORC from rebinding to the ACS after you've released from CDT1. And we fantasize about the idea that maybe that's because when CDT1 comes off, it sort of pulls ORC off the DNA at the same time. Total fantasy though.
The reason why I'm asking this, like 160 amino acid, the kind of, because of entropy, is going to be like ball up. But there seems to be a considerable distance for them to stretch.
It's much, so I will tell you, it's much bigger than it needs to reach. So we estimate you could do it with about 60 amino acids, and we've demonstrated that you can delete up to half of it without any consequence on that reaction. And in fact, it may even work a little bit better, which may be what you're alluding to, that if you get rid of a lot of the stuff, you don't have it all balled up in the middle. But yeah, and in fact, mutating, so I focused on I didn't really focus on it very much, but what we mutated is the part that's proximal to the N terminus. If you mutate the part that's proximal to the C terminus, there's absolutely no effect at all. So there it seems to be just a sort of a leash.
And I think like this N terminus is highly conserved.
They're not highly conserved, actually. The CDK phosphorylation sites are not highly conserved, but their presence is highly conserved. So that is where they are, moves around quite a bit, but that they have CDK sites is quite highly conserved. There is chunks, little, what some people might call slims, like short linear motifs, that are conserved. And those are what we really focused our mutagenesis on. So we do think there are short regions that are making critical interactions. I wish I could tell you more. There is a AlphaFold prediction that there is an interaction with CDT1. We spent a lot of time making mutants in that. We didn't see anything happening. So we were disappointed, and it's not in the paper. So.
All right, let's thank Steve again for today's wonderful talk.